having discussed about the processes which lead to the formation of zygote, now we are going to see how the embryo formation takes place both in monocots and dicots. And what you see over here is a plant which is having cotyledons, and what you have to pay attention towards in this particular diagram is, you can skip all rest of it, what you have to pay attention towards is this part of the diagram in both in that case and over here. This is a dicot seed, and as you can see, the seed coat is containing in it an endosperm and cotyledons, two of the cotyledons which have been formed from the embryo. Also, in the same case of monocotyledon, we have an endosperm and a single cotyledon which is present, as you can make it out from the diagram. We have this entire arrangement that has developed by further development which embryo has undergone. So, we are going to see how the zygote transcends to the stage where it is having the formation of embryo taking place. As you can see over here, here lies the embryonic root, and over here it is the embryonic root. Just for understanding which part of the plant we are dealing with and which aspect we are going to cover, I have used this particular description. And what we have next is the developmental stages of a monocot embryo. Now what you see is that supposedly we are talking about embryo formation in the case of rice. Rice is a monocot or any grass. It has formed a zygote in the beginning, that is a single celled stage. The zygote further divides and there is a formation of large sized terminal cell, while the other one is the basal cell, as you can see over there. Now this basal cell undergoes nuclear divisions and it could become a syncytial sort of stage, followed by the formation of cell walls, and there is formation of vesicular cell which grows in size as a result of the nuclear divisions. This vesicular cell and terminal cell, these parts you have to remember. Now the vesicular cell grows longitudinally while the terminal cell undergoes the division in such a way that the divisions are transverse. Alright, one is growing in this pattern and the other one is growing like this. Okay, and now as you can see over here, the lower part of the cell is increasing in the width. The division is such that the topmost vesicular cell stays over there. It gives rise to the new cells which add up downwards, while the lowermost cell, that was the terminal cell, it has undergone divisions. It has given rise to 2 cells followed by 4 cells, and this terminal cell gives rise to the cotyledon which divide and further. This cotyledon has a radicle, and the vesicular cells, the lower cells that vesicular cell form the suspensor. Now this suspensor is the joining site of radicle and cotyledon. Now moving back to the stage that we had seen, we see that the endosperm is there and the embryo is there. Now if you can recall what monocot embryo looks like, it is having an monocot embryo is called scutellum. You have to remember this. Okay. Now the scutellum will be having, it would be present like this, and the rest of the part would be cotyledon. Now this part is cotyledon supposedly, and this is scutellum. Over here you are going to find the shoot tip, the root tip. Okay. There is coleoptile and coleorhiza. I am sorry, the lower one being the cover of root would be coleorhiza. Over here we are going to find coleoptile, that is a sheet that covers the shoot tip. Now this shoot tip is known as plumule. Okay. Now this sheath covers the root tip, that is known as radicle. So what we are drawing over here is a cotyledon. What a monocot is having, the cotyledon components of a monocot seed we are considering. This is monocot embryo basically. Now how this monocot embryo has been formed, this monocot embryo is having a coleoptile, it is having a coleorhiza sheet like covering. This coleorhiza houses radicle inside it. We have a sidewards epiblast over here. Now, what are these arrangements? They are arrangements of the cells that we have to remember, and the rest of the part is having the cotyledon. How it has been formed? It has been formed by these particular divisions. The cotyledon is having the radicle. Cotyledon goes towards the side, radicle develops to the lower side, upper side there is differentiation of plumule. This suspensor and the vesicular cells, they form the formation of rest of the cotyledon. Now, the next developmental stage is of a dicot embryo. You are going to find two cotyledons, a plumule and a radicle. What you see over here is that the zygote divides, a suspensor, long suspensor cells are joined to a heart-shaped globular embryo, which later on forms that heart shape, and that from that heart shape you are getting two different cotyledons which are arranged. Okay, we have done the structure of monocot, dicot seed. If not, you can very well analyze that a pea is a dicot seed. The pea gets separated, both the cotyledons, and in between lies the embryonal axis, which is having plumule and radicle. Now, these two cotyledons are this developmental stage of an embryo, which are going to house the eating food material, and that would help in the development of the seed afterwards from cotyledon. And this is all you have to remember in terms of developmental stages of a monocot and dicot embryo. I am going to ask you to see this all over again. These are the stages, how the cells are dividing and giving rise to respective parts of the embryo. Over here what you see is there is formation of single cotyledon, because we do not have any heart-shaped globular embryo being formed, but because of formation of heart-shaped globular embryo, we have two cotyledons which are generated from the terminal cell. Alright, this you have to remember. Suspensor is what? Suspensor is the cell that joins this terminal and vesicular cell. Later on the cells keep on adding, and it is a stalk like structure that extends between this greater part and the topmost cell. Okay, so this is all you have to remember in the case of embryo formation. This part I just tried to make you understand that what is the difference between a monocot and dicot. This is a monocot that is having a single cotyledon surrounded by endosperm. That is why I made you I made you watch that first particular diagram. So I hope you get an idea about embryo formation in both monocots and